In this third chapter, we will study how the action potential arriving at the axon terminals evokes neurotransmitter release. Let's begin by observing synaptic transmission. Let's suppose we have two connected neurons. Here the presynaptic neurons in orange and the postsynaptic one in blue. The orange one is called presynaptic because it's located before the synapse, and the blue one is located after the synapse, so it's called postsynaptic. We record both neurons in whole cell configuration to record the activity of the whole membrane, and in current clamp mode to record potential changes. If we stimulate here the orange neuron, we record in response to the stimulation an action potential. And in response to this action potential, we record in the postsynaptic neuron, we record a small depolarization that we call, because it's a depolarization, we call it excitatory postsynaptic potential. Now, if we apply in the bath, here, we apply TTX, tetrodotoxin. This is the blocker of sodium channels. Of course, you don't get any action potential in response to the stimulation because sodium channels are blocked. And you don't record also a postsynaptic response. Now, if we use a bath devoid of calcium ions, 0 calcium, we still record the action potential in the presynaptic neuron because the action potential is due to sodium and potassium ions. But we don't get a response here in the postsynaptic neuron. Why? Do calcium ions play a special part in synaptic transmission? And so, what do they act upon? To understand the role of calcium ions in synaptic transmission, we work with the squid giant synapse, which is here. In this giant synapse, we see here the presynaptic terminal, the axonal presynaptic terminal in blue-green, and underneath is the postsynaptic element in white. And the synapse is beneath the blue-green, in between the two. It's a view from above. The result is as following. In A, this is the basal fluorescence of the presynaptic element. That's why we put it in green to show that it's the presynaptic element only. And when we stimulate, here it's the stimulation, when we stimulate the presynaptic terminal, we trigger action potentials, and in response to this action potential, we have image B, and to understand exactly what happened between the two, and we see here what happens during action potential initiation. We see that calcium entered the presynaptic terminal because we have here the fluorescence that tells us that the bond form increased, and it entered the presynaptic terminals with small microdomains like that. So this is because it's a giant synapse, and so you have many synaptic complexes and in this synapse, which is very different from the central synapses of the brain, where you have only one synaptic complex in general. So it's a peculiarity of the giant synapses. This is the Princeps experiment that allowed to characterize the role of calcium ions in synaptic transmission and to characterize that calcium enter the presynaptic terminal to the synaptic transmission to happen. So the hypothesis that we have now is the following. The action potential triggers the opening of calcium channels in the axon terminals. But what type of calcium channels do we have in axon terminals? And the white type, of course, of calcium currents. we should patch clamp and record the activity of axon terminals. This is very difficult in the brain, for example, with brain neurons, because the axon terminals are very small, about 1 micrometer in diameter. So we can use giant synapses, as we saw with the giant squid synapse, the calyx of health, which is in the auditory system of the brain, or the neuromuscular synapse, which is between motor neurons and muscles. We can also, this is a third possibility, record calcium channels in the soma, because the somatic membrane contains the same calcium channels as the presynaptic terminals, and it is usually what we do in the brain. So we know that there are many, but there are at least, we can say, 4 types of calcium channels that were called L, N, P, Q, and T. Now they are named CAV for voltage-gated calcium channels, and then they have a number after, which can be 1, 2, etc. And to understand which channels are present in the presynaptic terminals, we label these channels with antibodies, and what we see is that most of the presynaptic terminals contain N-type channels. Some types, also another type, which is called PQ, that we're not going to study. We're going to study the N-type, which is the more common form of calcium channels present in the presynaptic terminals. So we have now this new hypothesis. The transient increase in calcium, internal calcium, in the intracellular concentration of calcium, is due to an inward calcium voltage-sensitive current. Voltage-sensitive because we saw that we have to stimulate the presynaptic terminal to get this increase. And this increase is due to calcium entry through N-type calcium channels that are called high threshold because they open for a strong depolarization of the membrane. So let's test this hypothesis.